Today we're going to cover peripheral blood. We want to look at the gross description and the general properties of blood, and we want to look at the structure and function of the cells, red blood cells, white blood cells, and platelets of blood. Now, blood has diagnostic value. It's the most examined tissue in the body, and there's three basic types of information you can get from blood. One is you can identify the nature of the disease by what cell types are high in concentration. If it's a viral infection, you probably have more T lymphocytes. If it's a bacterial infection, you got more neutrophils. If it's a parasitic infection, you got more eosinophils. So when you go to the doctor to examine you, want to pull blood, that's the reason he does it. But the other two reasons, one, he can follow the course of the disease, getting worse, getting better. He also allowed a method to evaluate the effectiveness of a treatment. So those are three types of information gained from your physician pulling your blood when you go see him. And here we can see where blood cells are in the blood vessels, in the arms, the legs, throughout the body. And here we see the vasculature of the heart, thanks to body whorls. Now, connective tissue, blood is part of connective tissue, and here we can see connective tissue proper, connective tissue with special properties, and here we see hematopoiesis, and that's where blood resides right in through there, which is basically connective tissue. It has abundance of extracellular matrix, that is the fluid, and it maintains a logistic support and communication between tissues and organs like other connective tissue. However, unlike connective tissue, blood has no rigid physical connection among cells. It's not rigid. It's liquid connection among cells. Now, in blood composition, there's three major components. You have the plasma, which, and through there, the clearer part at the top when you spin blood down, heparinized blood down. And then they have a buffy coat, and that's where the white blood cells are located. And then they have a packed cell bottom, or your hematocrit, packed cell volume, is your hematocrit. Now, the liquid part of blood up through here, the plasma, is about 55% of the whole blood. In terms of the composition, it has water, has albumin in it. It has, albumin keeps the osmotic pressure, keeps fluid in there, and it neutralizes proteases. It also have globulins, antibodies, IgG, IgE, the different antibodies. It also has transferrin, which is iron, copper, zinc in there. It has chylomicrons, which are lipid components that are put into the bloodstream in the intestines and dumped out the lateral borders of the intestinal cells. Low-density lipoprotein for removing cholesterol as well. Continuing, it also has clotting factors. It has fibrinogen, prothrombin, thromboblastin, and thrombin, the different things associated with clotting in blood. It also has inflammational molecules, hormones, which direct cells' activity. It carries chemotaxic factors to affect the immune system, to attract them to any kind of infection that you might have. Now, blood plasma is different from blood serum. Serum is a yellow liquid from clotted blood. So you let the blood clot, then spin it down. This yellow liquid here is a serum, which is the serum is a plasma minus the clotting factors. The clotting proteins are spun down through here, so this is doesn't have the clotting factors, and you get serum without heparin. Now, in terms of blood, blood is a liquid tissue composed of erythrocytes, red blood cells, leukocytes, white blood cells, and platelets suspend in the blood plasma. And here we can see what happens. Blood that's in different blood vessels, blood comes in through here, return to the lungs, and it picks up oxygen. So you see the oxygen tension goes from 40 to 100, and then when it goes through capillaries again, it then drops off again back to the level that it has in the heart. So the function of blood is transportation. One word, transportation of cells and fluid. The red blood cells carry oxygen, picks it up in the lung, and distributes it in the capillaries, and carbon dioxide. It distributes in the lungs and picks it up in the capillaries. Now, the function of blood is transportation of fluids and liquids, as I said. The white blood cells are the immune cells of the body. The plasma provides nutrients for tissue, waste from tissues, protein to hold the water in plasma, albumin, hormones, and other inflammation mediators are in the plasma. It also has platelets. Platelets, we can see platelets right here, and here, and they prevent the loss of transportation, which is the function of the blood. So we'll talk about clotting of blood. Now, blood volume is about 5 or 6 liters, 12 or 13 pints. So when you give a pint of blood, you're giving almost 10% of your blood. And the reason you're able to do that is here we see a pie chart that shows the location of the blood, and the bulk of the blood is located in small and large veins. So these are reserves, so that reservoirs, so you can pull the blood out. You can pull out a pint and it not be a problem, or you can bleed for a while and not bleed out because you're pulling from these reserves. And so when blood goes through the body and it goes from the heart to the lungs, back to the heart, and then throughout the rest of the body, it travels, that is, different parts of the blood at the same time, travels 172,000 miles. It makes 960 trips a day, 170 miles of blood vessels per trip, and it takes about 60 to 90 seconds. So these are gee whiz figures. Don't worry about them. It is important that you have a reservoir of blood in through there. And so here we can see the blood vessels in the head, in the lungs. We can see these in the kidneys, and also the ones in hands and finger, thanks to body whorls. Now, the size of the blood cell is usually, for the red blood cell, is about 6 to 8 nanometers in thickness. Those are the red blood cells that we can see in diameter, I should say, from there to there. The white blood cells, leukocytes, are of different types. You got the neutrophil, which is about 12 to 15. So we're talking 2 to 3 red blood cells in thickness. So you can fit these red blood cells in through there. And about 60% of the cells are neutrophils. Eosinophils, in contrast, has a lower percentage, 2 to 4%, about the same size. Basophil, about the same size, but has a very lower percentage of these basophils that we see here. Lymphocytes, about 25%, and they are smaller than the other cells. Sometimes but it can be quite big. But usually they're smaller, more like red blood cells, maybe a little larger. And then we have the monocyte, which is kind of like a horseshoe-shaped nucleus, and then we have platelets. So those are the different components that we have. In fact, we call those formed elements, that is, the non-fluid cellular and functional, the cells that function in blood. You got the red blood cells, carry oxygen to carbon dioxide, neutrophils, phagocytosis of bacteria, eosinophils, parasitic infection, inflammatory response, basophils, release of histamine and other mediators, and that's what makes it itch. And then we have monocytes. Monocytes is a mononuclear phagocytic system. They become macrophages, could be fixed or random macrophages, random macrophages. Formed elements, as we mentioned, including the lymphocytes. You got T lymphocytes, which kill viruses. You have B lymphocytes, which generate antibodies against by producing plasma cells. These transform the plasma cells to T cells, or the killer cells themselves. You have the natural killer cell, kills tumor, virus-infected cells. Then you have platelets that is function in clotting of the blood. Red blood cells, erythrocytes, minute corpuscles that we can see here. It's produced in the bone marrow, and it's anucleated in mammals. It's anucleated. It has a non-functional mucus in reptiles, birds, amphibians, and fish. But even in mammals, it had a mucus at one time, but it was removed, and we'll talk about that a little bit later on. You can go from a natural state to a created state and back to a natural state without seeming to be much of a problem. There are lots of red blood cells in there, 2 to 5 times 10 to the 12th per person. Large. It's a larger amount at high elevations, if you live at high elevations. And that's why the Olympics team practices their sport in Colorado because it's a mile high. And that gives them advantage because they have higher hematocrits. Total surface area, 2,000 times total body surface area. So that's a lot of blood, a lot of surface area. Roulette is whenever these kind of stack like coins. Crenation is whenever if you change the osmotic pressure, it can go back and forth between these two. Now, there are words associated with variations in size. You can have small ones, large ones, shape, and also the shading, light or dark cells that you can have. Biconcave shaped, as you can see there, and it's brought about by proteins in the cell membrane, and also connecting to those proteins is spectrum that makes a mesh inside the cell membrane and facilitates this biconcave shape. If you have a hypotonic solution, it gets cremated, swell under water, and even pop if it's in water for such a long time. Sometimes you can drink too much water, and it can make your cells pop. Biconcave is important because it allows cells to migrate through, increases surface area, 2 to 3% greater diffusion of oxygen and CO2. Because of that, it's thin, so it doesn't have to, these gases don't have to travel far to get to the hemoglobin. It facilitates a passage through small things, small capillaries smaller than they are, and hemoglobin, 65,000 molecular weight protein with 4 polypeptide globular chains. Each has an iron containing a heme group that will attach oxygen and CO2 lightly so it can be attached, but then it deattaches as it goes into the tissue. We have myoglobin in muscle, hemoglobin in blood, as we can see, 2 hemes versus 4 hemes over here. I should say 1 heme versus 4, 1 versus 4. And so it looks like that this is just 4 of those things is what it looks like. In the erythrocytes can undergo sickle cell anemia. That's a condition for the person. And in that, you get the volate substitute for glutamic acid in 1 out of 114 amino acids on a beta change, and that causes a tetramer to be formed. You have these, and it makes a sickle cell. So as the fluid's pushing it, these tentacles kind of hang back, and that makes it a sickle cell. It reduces solubility of oxygen and causes crystals to form in the shape of these long crystals that make the cell sickle, prone to hemolysis and blockage of capillaries. That's the problem. Homozygous carriers of the HBS gene has it. Heterozygous carriers don't get it, but the homozygous do. Here we see consequences of mutation. Depends upon how it is, but in the case of sickle cell anemia, you get that mutation, and that causes a sticking of the different hemoglobin groups. As a consequence, you cause these linear structures, which is inside the cell membrane and makes these structures a sickle. Now, red blood cell reticulocytes come from reticulocytes. Reticulocytes is the precursor to the red blood cell, and it is what is released from the bone marrow. And within 24 hours, it becomes erythrocytes. So reticulocytes give rise to, and the difference is reticulocyte is blue. It has a bluish tint that we see here, here, and here compared to these others, as it has more ribosomes, and the membranes, the cell is a little bit larger, and those ribosomes are plucked out into spleen. Erythrocyte, lifespan about 120 days, then it's removed in the spleen. So the ribosomes are removed earlier, and then the cells removed in about 120 days. So here we can see cells coming down through here, different types. Ultimately, you lose the nucleus. The reticulocyte is released, and then you get erythrocytes in there. And the hemoglobin, a little different between the adult and the fetus. The fetus, the placenta has to capture the oxygen from the mother's blood and give it a carbon dioxide. So the hemoglobin is a little different during pregnancy than it is in the adult. And here we see these are megakaryocytes. These are different blood cells in the production of blood. If we look at that, erythropoiesis, and if we start with the most mature, these are erythrocytes, and these are reticulocytes. And then if you go from reticulocytes to orthochromatic normoblasts, and this is the one that dumps its nucleus. As soon as it dumps its nucleus, it becomes a reticulocyte. Before that time, we have a polychromatic erythroblast. That is polychromatic. The cytoplasm is kind of gray, like the mature one, and then blue, gray and blue, combination, polychromatic, more than one color. And then we have a basophilic erythroblast is through here, maybe fractured face somewhat, blue cytoplasm, and then pro-erythroblastic, one that doesn't have a fractured face is more. But all these cells have a spherical nucleus or no nucleus at all. And so if we look at the orthochromatic erythroblast, you can see the nucleus is being a lot of heterochromatin being condensed. Few ribosomes are located there, but the hemoglobin's in there as well. And in fact, if you looked at what that is high density of ribosomes and also hemoglobin. So hemoglobin's there, machinery or ribosomes here. So you always start out with machinery and end up with product. Machinery being ribosomes and the product being hemoglobin. And here we can see that reticular protein is discharged at a reticulocyte. So the nucleus is discharged. Here we see the nucleus is discharged from the orthochromatic normoblast to the so once the nucleus is removed, it's a reticulocyte. And so if we look at, again, the most advanced ones and then go back in terms of time, we can see this would be the red blood cell, this would be reticulocyte, and then this would be orthochromatic erythroblast, and then this is a polychromatic where you get a sea of ribosomes, also lots of hemoglobin where you got more hemoglobin here, and more machinery or ribosomes in the basophilic and the pro-normoblasts. If we look here again with the more advanced erythrocytes, you see the bluish tint, you see the ortho popping it out, polychromatic is more varied in size, but you can see the pink and the blue combinations becomes more fractured face, but becomes a little less dense. As we go through here to the basophilic, you got blue cytoplasm, and then again blue cytoplasm in through there for the pro-erythroblast as well. So in terms of what changes is the nucleus goes from a more euchromatic to a more heterochromatic, a larger one to a smaller nucleus. The cytoplasm goes from dark blue to polychromatic to mostly just a light blue tint, more red, and then finally red. So those are the combinations that occur for the different named cells. And here we can see where our erythrocyte is entering the bloodstream, as you see here and here. So this is in the bone marrow, and we can see this is a blood vessel in through there. And the blood cells are produced outside the bone blood vessels, but then they have to migrate through. And you can see where this one cell is squeezing through. Now, the granulocytes are of different types. You got the neutrophilic granulocyte, eosinophilic granulocyte, and the basophilic granulocyte. So all these are granulocytes. They have a lobulated nucleus. There's also non-granular leukocytes, and that would be these other cells here in green, the lymphocytes and the monocytes. And the monocytes give rise to macrophages. And here we can see in the different cells, the neutrophil, eosinophil, and basophil, the different types of enzymes that are in there. In the neutrophil, you have both specific and aerophilic granules. This is more like the lysosome, and this is the one that kills the bacteria in through there. In the eosinophil and basophil, their granules are the specific granules. They don't have another set of granules. Here we see where white blood cells eating yeast, and here's where they have digested bacteria in through there, as you can see in the specific granules that would be killing the bacteria that was phagocytized. Here we can see more of the eosinophilic granules. And then in terms of advancement, the cells, as they mature, the nucleus is lobulated in these granulocytes, go from a spherical one, a oval one, indented, and then finally the nucleus is lobulated. It's not lost, it's just made into lobes. And you go from ribosomes to granules, more granules, and then finally the mature granules in through there. It takes about 6 hours or so to do that. Now, in terms of maturation, the cells become more mobile, they're more active, the features are more predominant, more likely to adhere to blood vessels than the younger ones, and more pliable. So these are the maturational characteristics of these cells coming out of the bone marrow, going into the blood. So it comes from out there, and there's one blood cell, another red blood cell, another one there. And so you can see that that it is, in this case, it looks like it's coming out of the blood, going into the area, kind of like this, where this was the blood in through there, and then this is the infected tissue that it's going to. So here we see the different cells, spherical nucleus, which is more on the red blood cell series, or the lobulated nucleus that's going to be on the granulocytic series. If we look at the granulocytes, again, with the most mature, we see a neutrophil there, kind of gray cytoplasm, and the nucleus is in lobes. It's not multiple nuclei, it's all one, just in different lobes. And then, so that's a neutrophil, and then the neutrophilic band cell or stab cell is kind of like a horseshoe shape, and then the neutrophilic metamyelocyte is more indented, more like a kidney. And then a neutrophilic myelocyte, that was myelocyte doesn't have that indentation, and also the cytoplasm is varied. It's blue, and there's some granules in there as well. Monocyte is less dense nucleus, and that's what changes. The nucleus gets less dense and lobulated. The cytoplasm goes from very blue to less blue, more mature cytoplasm, kind of grayish as you see. So those are the characteristics of those. And in bone marrow, we can see blood vessels in through there. There's granules that you see in the developing cells that we can see in there. And so here we see neutrophils, and this is a neutrophil here, a lymphocyte there, another neutrophil which has two types of granules, the kind of bigger ones, and then these dumbbell-shaped ones are the specific granules that we have. So this is the scheme again, go from the most advanced to the least advanced, lobulated nucleus, less lobulated, and then more oval. Cytoplasm mature with granules, mixed with granules and rough endoplasmic reticulum, and then with ribosomes, and then more and more basophilic cytoplasm as you get more ribosomes in a more immature state. And here's another blood vessel in bone marrow, and we can see where one of those granulocytes are squeezing through from the outside going into the blood vessel. Now there's two types of leukocytes. There's granulocytes, which are neutrophil, eosinophil, and basophils, and then there's non-granulocytes, which are lymphocytes and monocytes, as we mentioned before. 20 to 30 billion in circulation at any one time, and white blood cells, 55 to 60% of the cells, and there's a good amount of these. In case of infection, you increase the amount. You can also see the segmented thing. This is actually the bar body, and that occurs whenever the X chromosome, in the case of the female, has an extra one, that bar body. Males don't have it. And then we have eosinophilic granules in there, acid phosphatase, hydrolytic enzymes, primary lysosomes, and then there's also basic protein, which is responsible for killing the bacteria. Neutrophil band cell, as we can see there, if you have an infection, you will cause these precursor cells to be released in higher numbers. Opsonized is whenever antibodies facilitates phagocytosis. So basic cellular function is a random movement. They move out of the bone marrow. Chemotaxis, they're attracted to wherever a bacterial infection is. Phagocytosis, they can phagocytize it, and then they kill it with their specific granules attaching to the vesicle that has phagocytized it. Eosinophils, you can see the bright red granules, but it's also lobulated, as we can see. It only lasts for a few days, and you have higher concentration of these if you have parasitic worms. Common in the lamina propria of the GI tract, respiratory tract, attracted by chemotactic factors. If you need more, they'll pull something there to go where a parasite would be. And the role is parasitic disease and allergic response. Here we see an eosinophil with electron micrographic view. Has these big granules, which we can see at light microscopic level, but what we can see at EM is that they have a crystalline core. So they have a crystal structure inside the core there. The only type of white blood cell has that. Eosinophil and basophils, red granules and blue granules. And the basophil is least common, doesn't have a crystalline core, but it have metaplasia. And that is, it has heparin, and whenever it stains different colors. And so you have a combination of different colors within the same granules. May be induced to degranulate, like the mast cell, specific granules, IgE molecules on its surface, and also 20-fold in a person with hay fever, allergic response, or something. So you can have a lot more of those if you have hay fever or something. Basophils has histamine, which causes the itching and increased vasculature, so it's edema, so it swells. Contagious basophilic hypersensitivity. If you use a certain window, rebuck window, which is you scratch your surface, put a cover slip over it, and that would be the window, and then the basophils will migrate to that, indicating that they not only can move, but they can move into a certain area where chemotaxis would be calling them. So here we see neutrophils, monocytes, red blood cells, platelets, another monocyte right there. And here we can see a nice display of the different cells. Eosinophil, big granules, big blue granules in the case of neutrophils. A neutrophil, the cell doesn't lay down. So in eosinophil and neutrophil, the granules lay down the side. They don't really, there's one right here, but not many over the nucleus itself, where the basophils, the granules won't lay down the side, and we can't see the nucleus that well because of the granules on top. Blue granules, big ones, red granules for the eosinophil, and you have the two types of granules in the case of the neutrophil, but they're smaller than these other two. Monocyte, kind of horseshoe shape, kidney shape, and the lymphocyte, which has a lot of nucleus and not much cytoplasm there. And these are derived from bone marrow. And so you have the B cells, which give rise to plasma cells, and you have these cells that have the different groups. You have a helper cell, suppressor cell, killer cell, and then you have a memory cell that remembers things. That's how you can be immunized, because you have cells that remember what they were exposed to. So when they're exposed to that again, they attack it quickly. And that's the whole premise of immunization, is that the body has already built up a resistance to something that they're going to be, and a defense mechanism for whenever they're exposed to that again. B cells, you get plasma cells, antibodies, memory cells, T cells. T cells recognize specific antigen, recognize the surface on the cell. Memory, cytotoxic helper cells, as I mentioned to you before, and suppressor cells dampen the response. Don't kill the patient because you are mounting immune response is kind of what it is. So here we can see lymphocytes here and here, and the lymphocytes give rise either to T cells or, in this case, plasma cells that produce antibodies, or this is a T cell. So this cell gives rise to this one, which is the same as that one, which has a lot of cytoplasm, and that's rough in the plastic reticulum. But we don't see granules because it has a constitutive secretion. As soon as it produces it, it's gone. It doesn't store its secretions. If you need more antibodies, you need to get more plasma cells. So, and the T cells, we can see what happens when the cells see their antigen. They respond by increasing the DNA, increase cell division, increase in their immune response. And here's this margination. This is marginating, where a white blood cell is attached to the endothelium and it's going to migrate through. So it attaches to it. Normally they just ride by, but as it attaches to it, they can slip through here, out of the blood vessel into, so it goes along there, attaches to it, slips through the endothelial cells to get out, to mount the response on the outside, margination. Now here we see plasma cells, several here. See the cartwheel-shaped nucleus, spherical nucleus, and then lots of cytoplasm there, and that cytoplasm has endoplasmic reticulum. This is bone marrow. We can see blood vessels running through there, and the big cells there are megakaryocytes. So here we see a lymphocyte squeezing out of the bone marrow. There's another one there into the lumen. Leukocytes, humoral immune response, not cell-mediated, but humoral. It's the B cells, the plasma cells to antibodies. The cellular response is directly where the T cells kill things. And so cell-mediated as opposed to humoral-mediated response, you have memory cells, helper cells, suppressor cells, and the killer cells. They interact with the macrophages, cytotoxic phagocytizing mechanisms, and they produce interferons that are produced by T lymphocytes capable of inhibiting replication of certain viruses and capable of reducing the multiplication of parasites. Here we see platelets. This is an endothelial cell. We can see platelets have structures inside them. Platelets here in the regular blood smear. And here we can see the megakaryocytes in through the bone marrow, which gives rise to the platelets. Here's another big megakaryocyte there, and those are the ones that make the platelets. Here we can see the nucleus of one, and if you look at an ultrastructure of one, you can see there's a series of little vesicles in here, as if this is ready just to pinch off as a platelet, pinch off as a platelet, like that. Now what happens is megakaryocyte stays in the bone marrow, but it sends out projections in the bloodstream, and then those pinch off as platelets. And here you can see where that those long projections are already going to pinch off at platelets. Now, in terms of clotting blood, and these are 5 steps, it'd be very handy to understand these. One is a primary aggregation. That is, the platelets get attached to wherever you have a cut. And then there's secondary aggregation. So alpha and delta granules induce further aggregation. So you get some started, and then they call for recruits. And then you get a blooding cascade of plasma proteins to produce the clot, or the thrombus is what it is. So that contains enzymatic activity. And then from there, you get a clot is retracted. So the clot is retracted. At that time, it's called actinomycin. ATP is involved. And then finally, the clot is removed. So those are the primary aggregation, secondary aggregation, cascade of proteins that make the mesh, and then the retraction, and then finally removal of it. Now, platelets have a role in stopping the bleeding, is what they do. They produce serotonin, and serotonin itself is a vasoconstrictor. Also produce thromboblastin. And so here's prothrombin, thromboblastin from them will make thrombin. Thrombin makes fibrinogen and fibrin. And the fibrin is what polymerizes and produces the plug. You can see where the fibrin is. So serotonin, fibrin, to cause constriction, to block the outflow of fluid. And so here we see there's a whole group of things not to be concerned about here that's going on enzymatically, resulting in a mesh of cells that clogs up an outlet. So in terms of platelets, they have many metabolic function of whole cells, but they don't have a nucleus in mammals. When exposed to damaged cells, they are sticky and cause aggregations to stick one another. Release ADP from ATP inside platelets, which attracts other platelets. And so the ADP attract other platelets, and it produces thromboblastin, which has the cascade to produce fibrin, which forms the net. And here we can see the net, which is formed inside there, the basis of the clot. So in summary, our platelets, hemophilia is a bleeding disease, and that's inherent single deficiency of any or the 12 clotting proteins that are involved there. So this person will bleed. Also, if you take aspirin, you will bleed more often, but not like a hemophilia will. So there's anucleated in mammals. We don't see a nucleus. It has different zones zone, halomere, granulomere. Halomere is on the outside, granulomere is on the inside. Granules contain serotonin for vasoconstriction, platelets produced by megakaryocytes, bone marrow, and at least 8 to 11 days for them to last. Next time we're going to talk about epithelium, which is one of the 4 basic types of tissue. Epithelium, connective tissue, muscle, and nerve. Either that or we're going to go to Washington, D.C. Here we see the Capitol, and there's Washington Monument right there. That little dome back there, a little bit there, is the Library of Congress. And this is Hope Diamond and in the Museum of Natural History, Arlington Cemetery in Arlington. And this is on the grounds there, heading between the Washington Monument and the Lincoln Memorial. There it is again, about in the same area. There's a Washington Monument, and this is a view from the Vietnam Memorial that we see there. We want to thank many books whose pictures we've used to illustrate the various components we have there. I did not originally make these things. They came from these various books that we thank for their contribution to education. Thank you.